Hi, so I'm Anna, and I'm going to teach you about using a DNA-based scaffold, DNA-based delivery particle in particulate vaccines. Um, so basically, using a DNA-based delivery particle in particulate vaccines to induce a strong antibody response against viruses is a technology that can create new ways to prevent the threat of new viruses and also allow for new developments in medicine for public health. And to understand this better, you need to know what a particulate vaccine is. So basically, a particulate vaccine contains tiny particles of dead or weakened microorganisms, which the immune system will recognize as foreign and then initiate an immune response to. So for you to better understand this, it's important to understand some key points about the immune system as well. So the immune system basically has two branches, the innate and the adaptive immune response. So the innate response is essentially like a rapid, non-specific response that has no memory at all. So basically, every single time that you get infected with the exact same strain of one virus, or you get the same cold over and over again, your innate immune response will respond the exact same using the exact same cells every single time with the same magnitude. Nothing will change. And then this is the virus or like whatever it's encountering that's not like from you. That's a non-self molecule known as an antigen. So like that could be like a bacteria, a parasite, virus, anything like that. So meanwhile, the adaptive immune response is a slow-acting, highly specific response with memory. And it's capable of providing a response with much higher magnitude when it encounters that same antigen for like a second, third, fourth time, whenever, because it'll have memory. Okay, so in this one, I'm going to be focusing on the adaptive response, like in this presentation, because this topic is super complex and it just applies more to the adaptive side, and it's, I think, a lot more interesting in my opinion. And it's also kind of what the journal that I read, like all of this about, it's what it focused on. So this is going to be mainly focusing on the adaptive response, but both the innate and adaptive immune responses are triggered in an immune response to a vaccine. I'll just be focusing on the adaptive. Okay, so the adaptive immune response is made up of two branches, humoral and cell-mediated. So the humoral response mainly involves B cells, and B cells are able to differentiate into plasma cells, and then plasma cells will secrete antibodies that will fight the antigens to protect us. On the other hand, the cell-mediated response is made up of T cells, which are able to kill virally infected cells and cancer cells. So here, obviously, the virally infected cells that will apply here, but the cancer cells won't. But I think it's important to mention that they're able to do both. And they're able to do this upon being presented an antigen by another cell that's known as an antigen presenting cell, or an APC. So the T cell essentially can't recognize non-self molecules, the antigens, on its own. It needs to be presented that antigen by another cell. So T cell-based immunity is strongly elicited by vaccines, such as the mRNA vaccine. However, particulate vaccines are very good at activating B cells. And B cells, remember, are the ones that make plasma cells, which make antibodies. And antibodies are what fight against the antigens that like surround it, and then that makes it easier for other cells to see it and eat it and kill it. So the particulate vaccines are very good at activating B cells, which means they'll provide strong humoral immunity, which is essentially strong antibody-based immunity. So after the initial exposure to an antigen, both T cells and B cells will make memory cells, because both B and T cells are part of the adaptive immune response. So they'll form memory cells that are specific to the antigen that like they were formed for. So like when the cell, like let's say when the T cell encounters the antigen that it was presented by the APC, it'll make memory cells that are specific to that exact antigen. So if you get a different like bacterial infection, they won't respond to it. But if you get that same one again, they will respond to it. They'll be reactivated and they'll recognize that same one and they'll give a stronger and faster response in that secondary immune response. So conventional particulate vaccines use protein-based scaffolds, which is different from what I talked about in the beginning. I said that I'm going to teach you about using DNA-based particulate vaccines, and now I'm talking about protein ones. So basically, this is just the conventional form of it. The DNA-based scaffold is pretty new, which is why I wanted to teach you about it. I feel like it's really interesting. But the conventional particulate vaccines, I think we need to go over that first so that you can understand it a little bit better. They use protein scaffolds to present the viral antigens, and viral antigens just means like a virus, while mirroring the structural features of the pathogens. Pathogen, again, it just means like the virus. To be able to initiate an immune response. And the thing is that the protein scaffolds distract the immune system's attention from the target antigen and cause the immune system to produce antibodies that will target both the protein scaffold and the antigen that it's presenting. And that's not what we want. We would want it to only be attacking the antigen and not the protein scaffold. So this means that the immune response won't be as strong as is like desirable. Moreover, if a single person receives a vaccine that uses the same protein scaffold twice for two different viruses, in that second vaccination, their immune system would likely respond immediately to the protein scaffold because it will have made memory cells for that protein scaffold, and it'll recognize it as an antigen that it needs to like fight, which will make the immune response weak to the actual target antigen, because instead of just focusing on the antigen and attacking it, your adaptive immune response will be responding to the scaffold as well. So it's like basically you're dividing its attention into like a thing that holds the antigen and the antigen instead of only focusing on the antigen. But DNA-based scaffolds in particular vaccines are something new that can maybe avoid this. So basically, researchers have been creating DNA-based scaffolds using DNA origami technology, which allows them to control the structure of the scaffold and attach different molecules to any specific location on the surface of the scaffold. So this scaffold was found to be immunologically silent, because researchers saw that in their new study that the spike protein's receptor binding protein was replaced with an antigen from the original coronavirus strain. So basically they're doing like a new study in this article with COVID. And so here I'll be like talking about COVID a lot. Basically that's like the virus that they're going to be testing the DNA-based scaffold with. So basically, after this vaccine was given to mice, which are commonly used for like this type of testing, because they have very like similar genomes to people, it was observed that large amounts of antibodies against the spike protein were produced and none against the DNA scaffold. So the spike protein that I'm talking about, that's essentially like the coronavirus. And what I just said there is basically meaning that like the DNA-based scaffold that they made for the particulate vaccines, like the thing that's going to be holding the antigen, they were able to make it in a way that like using that DNA origami technology, they're able to manipulate any like molecules that they put on any place on that like scaffold. And they made it so that there were no antibodies produced against the DNA-based scaffold and only against the antigen, which is here the spike protein. And that's good. That's exactly what we want. So basically the adaptive immune response is not being distracted here. So that's super cool because I don't think we've found anything like that. So on the other hand, a vaccine that used a protein scaffold covered with coronavirus antigens produced a large number of both antibodies against the coronavirus and the protein scaffold. So that just shows the difference. They basically did the experiment like on two things, like with the coronavirus, like spike protein with the protein scaffold, and then the DNA-based scaffold with the same like spike protein of the coronavirus. And in the DNA one, the DNA-based one, there was antibodies produced against only the spike protein, and then in the protein-based scaffold, there was antibodies produced against both of them in like large amounts, which is like not good. So by presenting viral antigens on a DNA scaffold, the vaccine promotes a targeted immune response against coronavirus, like specifically, and that can potentially offer long-term protection against the virus. And also by putting many copies of the antigen on the DNA-based scaffold, it mimics nano-sized viruses, which cause the B cells to elicit a strong immune response, producing many antibodies in super high quantities and good quality too, due to the way in which the antigens were presented on the DNA scaffold. So that's really cool, I think. Okay, so basically some research findings from this. Basically, researchers have been creating DNA-based scaffolds. Okay, sorry, I forgot to delete a slide. So basically, ignore that one. And oh wait. Okay, yeah, no, ignore this one. So I'm gonna continue here. So basically, the impact and significance. So the discovery of the DNA-based vaccines really like actually fills an important knowledge gap in vaccine development and in immunology by overcoming the limitations of the protein-based scaffolds. So people such as like vaccine developers and physicians could see this finding as an opportunity to improve vaccine effectiveness, since the DNA-based vaccines create more targeted and more specific immune responses. So the DNA-based vaccine is able to cause a long-lasting antibody response, which makes a way to develop vaccines against a wider range of infectious diseases, such as like the flu or HIV. And this technology can also create new ways to prevent the threat of new viruses. So overall, DNA-based scaffolds can act as a substitute for protein scaffolds, and that's a huge improvement and a huge finding in vaccine research. And it can allow for new developments in medicine for public health. And I think that's really cool. So after that, I have my task. I don't know if you wanted this on the slides, but I included it on the slides and then the checkpoint 5, like it's like if you scroll down there, I put it on there too. But basically, I just thought it might be cool to like make a immune system diagram, like just a basic one, like with the things that like I went over. And I have the steps on there, and like I thought maybe just like mapping out like, oh, like immune system, there's like adaptive and innate, and then I didn't really talk about innate, so you could just stop there. But then like for adaptive, you could be like, okay, there's humoral and cell mediated, and then humoral is like B cells and like plasma cells and antibodies and stuff. And then T cells, you could be like, oh, kills virally infected cells or like cell mediated. I don't know. I just thought maybe like making like a diagram or something would be a good way to like better understand it in like a easy way, like using stuff we have at school and whatever. So yeah, basically that's it. That's my presentation. Thank you. I hope that I was able to teach you something new. Thank you.